is the Institute of Applied Science and Intelligence System. We are in Naples, South Apart, 200 kilometers south of the capital Rome. So let's talk about this labor-free tomography for our intelligence, because we are using some AI for this one, and also for application phenotyping and other things in tomography. But before to do that, just a few seconds, just to say that in addition to this research line, we are working also on the biointerface between cell and materials. These are very recent papers in which we have photorefractive material that can affect the cells on the surface. Or, for example, we are working on the possibility to have also labor-free for histopathology tissue analysis, and also for sensors, which says by using a patented system about pure electric jet printing system and so on. So this is a summary of the talk. So I will start with the microscopy, then I will go direct to the QPI through the digital holographic microscopy, and then I will show some application in flow cytometry, 2D and 3D, and I will talk to you about this problem about lack of specificity of interferometry technique. It is very powerful. You know, interferometry probably is the most sensitive system created by humankind. It is used for gravitational waves, so you can get a lot of very sensitive data about the accuracy about the system by means of interferometry. The problem is how to use or exploit this kind of information. So I will go to how the system is doing, and also I will give an overview of applications. So in general, we know biology is a fundamental tool for inspecting biological material from vegetal world, also biological animal, and in our case, since usually there are some lenses more or less complicated that you put inside the microscope, you can look at it and see all the material characteristics. So the first question is 2D or 3D. And the second aspect that we know, we are aware now that essentially probably 95% of microscopic pressure, okay, right now. So everything is computational now by the digital means. And so the question is, a cell is a three-dimensional animal, so you should, in principle, have a three-dimensional microscope to look at this. But if you see overview of the state-of-the-art of technology in microscopy, you can see confocal to photon microscopy, light sheet, and we propose holographic tomography. But essentially, all the first three are based on fluorescent microscopy. Okay? So you need a chemical agent to visualize the object. Now we are proposing instead the possibility to use a labor-free, just to change completely the paradigm, just to avoid all the problems connected with the fluorescent analysis. But let me go back just one step back about history between microscopy and Nobel Prize. And so if we go see to this picture, essentially for optical microscopy, not for scanning electron microscopy, we can see that essentially in 1953, Zernaik just is a very little improvement. Now we can consider now this slow. He sold the Nobel Prize because it was revolutionary, because it was the first time, because usually the cells are transparent. They are, the most content is water, so you cannot see through a bright field microscope. And this was a revolutionary step, a challenging step, because it was possible for the first time to see with a simple microscope to see the edges and also the contour inside the cell. Then the second one was in 1971 by Gabor. That is accounted for the invention of holography, but now I'm talking about digital holographic microscopy. That's something that came the first step from holography, of course. And then we can see also the Nobel Prize in 2014. The fabulous aspect that we have a super resolution, and probably the most knowledge that we get as a humankind about biology is due to this important step. But however, the first comment, this Nobel Prize was awarded in chemistry, not in physics, even if microscopy is a branch of physics, as we know. And because you need here to have a chemical agent that mediates the object, and so you have this not direct imaging through this fluorescent analysis. And this can be a big issue in principle. So one of the inventors this is a paper just appeared the last week. So of course, they are aware. Okay, I cannot offend them. They are aware that fluorescent microscopy has a problem. You know, the problem, a lot of advantages, but also a lot of limitation for that everybody knows about this kind of things. So I will start to talk about this QPI, the possibility to use quantitative phase contrast imaging. So while Zernaik started with the qualitative phase contrast imaging, and essentially you see this fibroblast, this time lapse overnight. You can see these nice color images here, which you can guess probably the nucleus here. What's happened is a coherent wavefront passes through the cell, so it accumulates some retardation, some delays due to the thickness, the physical thickness. That means a mix between the refractive index and the physical thickness of the object. So the mixing of these two make this shape. So it takes a sort of a imprinting of the wavefront. So we are evaluating this shape of the wavefront passing through the object. And essentially, which is the main difference? Here, if you use an engineering a fluorescent probe just for the nucleus, you can exactly say, more or less, if you negligible the errors, that it is the nucleus. Here, you can guess that the nucleus probably is here, the thickest part, but you cannot have an exact boundary of the nucleus in this case. And so, we know this is very important. Why it's important? The laboratory is interesting for this kind of biology or clinical and so on. For someone that would like to have more detail about digital holograph, there are many books. Among them, one is this one. The second aspect of the modern biotechnology is the single cell. There is a fabulous paper in which he explained that the single cell is the new frontier for the imaging. So independently from this huge statistic that you can do, now it's important to look also at the single cell. And to have an answer with you know that the state-of-the-art technology are flow cytometry. So there are essentially two classes of flow cytometry, just a simple scattering, fluorescent or non-fluorescent, in which you just make a laser beam interacting with the cell flow inside, very high throughput in this case, and imaging flow cytometer. This is the high throughput is not so good like scattering here, because you have to capture images while the cell are flowing inside the microfluidic channel here. But again, they are based on the fluorescent analysis. This is the standard how to put for scattering flow cytometer, and this is the standard how to put for multi-channel image flow cytometer, which you can engineering different fluorophores, and then you can visualize different part or subcompartment of the cell, just visualize. Of course, you are still limited by resolution of your optical microscope and so on, and there are a lot of applications from marine pollution, animals, and biology, and so on. But again, the main problem is that it's based still on fluorescent microscopy. So we can say that fluorescent microscopy has an excellent specificity. Like you engineering them, the specificity, what you would like, you can visualize. It's a single cell, it's high throughput, but it requires staining. That's the main problem, and also is limited at the state of art of technology today to the two-dimensional analysis, not 3D for this kind of flow cytometry. So essentially, we believe that probably the microscope, the ideal microscope of the future, will have all these features simultaneously: stain-free, single cell, 3D, quantitative, and high throughput. And maybe by means of the modern tool of AI, all the computational aspect maybe can be reached or at because the first step has been done. So let me start with some of the results. Let's see QPI flow cytometry in 2D, just limited to the cell flowing here, and just to see and evaluate the bidimensional phase contrast map, quantitative phase contrast maps of this kind for liquid biopsy, CTC, searching the blood and so on, or phenotyping. Essentially, the system work like this. We have a very simple plastic chip, cell flowing inside. We have the coherent beam that is with the camera that is capturing the things. Few examples. Labor-free assessment of drug resistant ovarian cancer cell. Just we were able just to distinguish different kind of cell. This is a typical recording of digital hologram movie. You can see fringes, okay, maybe. And this is the raw hologram movie, and this is the face map that the quasi real time we're able to capture. This is something that is very nice and colorful, but has no meaning unless you make a full quantitative analysis. This is a very sensitive measurement here. But we are not looking now for detecting supercompartments. We are looking at this rich content, high content, face contrast map, just to say, are we able to distinguish, based on this high content face contrast map, different kinds of cell for phenotyping? And the answer, limited for our things, was yes, because we think this, we analyze with the standard machine learning and just putting inside many parameters. We included also the very sensitive interferometric phase quantitative analysis, and we were able to distinguish to separate population of the cells that are resistant to the drug and so on. Other aspect, just to make this a project that finished just a few months ago, in which we try to estimate which is the right dose for a personalized X-ray therapy for tumor, because this is very important. Right now, the state of the art is very difficult, because you use some dose, but you are not sure that you are killing also good cell or only the disease. One more thing is phenotyping, just for for liquid biopsy, we make a phantom mix of different cells, and so we try to, by machine learning, to extract any of this activity, and so we have monocyte versus neuroblastoma, just to see, because of course if you make CTC for liquid detection, for liquid biopsy analysis, we have a lot of components inside the blood, but of course the cell that can be confused with the circulating tumor cell, for example, monocyte for shape and tints. And so neuroblastoma, different class, subclass of neuroblastoma, and say again, that's differentiating and phenotyping different many subclass of neuroblastoma. Then it seems that the high content, the parametric content, through the machine learning is able to phenotyping this kind of stuff. So we are trying, as I said before, this liquid biopsy. You can see here a typical neuroblastoma. You can perceive probably the rotation of the cell flowing along the microfluidic channel. And what's happened, we made a lot of experiments. For example, for cell classification by using a low amount of the data, or hiding by unbalanced data set and other things, just to see which is the most practical way to this. Or, for example, by having clinical information together with the result of the machine learning, just to extract the best answer for our question, in which are the possibility to recognize again different cell lines in different conditions. But now, our ambition is to go to do everything in 3D, so pass from 2D to 3D, real 3D analysis of this kind of stuff. And of course, what do we expect? We expect just to classify and to detect and quantify volume, density, and recognize the subcompartment cell inside the cell by means of a refractive index, density of their own density inside the cell. So morphology and density. And I will show how we're able to detect some of this subcompartment, also lipid droplet, vacuole, and exogenous material inside such kind of cell. So essentially, our ambition to say, or our answer, our question, we don't have the answer yet. But is it possible by flow cytometry in quantitative way, but it's a huge amount of very sensitive data, to have a single fingerprint for the cell? Maybe this is challenging. This is our ambition to demonstrate the possibility that eventually that can be done, because that can make a revolution, of course, in labor-free microscopy. So how it works? Essentially, quasi real-time detection of the cell. So we are able to detect also rotation of these cells in a standard tomography system. The patient is at rest, and the X-ray detector they rotate around. Now we are at rest with the laser and the camera, and the cell is engineering the fluidodynamics just to rotate the cell. So inside the field of view, we have many rotated poles of the same cell. Then we put all together thanks to the three-dimensional feature holography. Okay. Okay? We don't care if the cell is at different depth, like in fluorescent analysis. We can do everything by computation analysis, just to put all the poles aligned, all the angle detected, and so on. And in this case, essentially, while in this case red blood cell is rotating, we are capturing different rotation. We are able to track accurately the angle. I will discuss now the detail about this one. But essentially, this is the first tomograph we made for a red blood cell. This is quite not meaningful information of phase map, but at the end, you can reconstruct. This is, and in principle, you can measure here the content of hemoglobin in each voxel of this red blood cell. Okay? Not only the average in a single blood cell or the average between 1,000 of cell aggregates. But the problem is the lack of specificity. That means specificity is a huge problem because we have a lot of data, but we have no information how to detect the different objects. We try to make some comment in this paper some time ago. But essentially, we believe that essentially digital holography and flow cytometry have all the characteristics that affect this problem, and so we are focusing our attention to solve this kind of a problem here. And so, starting from here, we would like to have a full three-dimensional quantification of the single cell, like in this case. If you just project the topograms, just with a different threshold, this cloud of voxel is completely nonsense because you never find a threshold that visualizes the nucleus or the mitochondria or the other things, just a confusion, things like that. And so we started to work on the possibility to use statistical method. There are two possibilities. Statistical high, a group in Korea, they started with the high, but of course you have to feed a neural network to do that. It's very. Instead, we use this procedure to statistical things, and essentially, I will go straight to the result. And we found a way just to have to accumulate all the voxels that, for sure, or with a robust statistical criteria, can belong to the nucleus, just to identify the nucleus inside the cell. As you know, the nucleus ratio between nucleus and cytoplasm is important in this case. So essentially, what's happened that we pass from this vision to this vision, which we have quantification of the nucleus in size, volume, location, and also measurement. And then we made the second step, in which we were able also to identify also, I don't know if you can visualize here, also nucleoli inside of the nucleus. That means that we are scaling down, try to find a good, going down to visualize everything. So we made some experiments just to make, for example, confocal microscope validation of budding SSL, to see the process. And here you can see that validation can be quite good in many things. Then we added some AI strategies just to improve it, to boost our analysis. For example, just speeding up the process, data compression, because we have a lot of data in terms of the tomograms of this object, the phenotyping, and so on. So just to jump all the steps between the raw hologram and to the tomograms, or for example, to compressing all the minimizing the lossing by compressing the 3D data just in a one-dimensional string of data, just my mix of Zernike polynomial. This Zernike polynomial is very famous for aberration calculation for lenses and optical analysis. As you see, we were able to Zernike to save about more than 95% of the space. But we were able also to visualize exogenous structure. In this case, graphene nanoparticles that accumulate inside uptaken by the cell at different concentration. You can see very small aggregates of graphene nanoparticles that are accumulated, so you can visualize exactly where they are, which is the volume, and so on. Or even in condition of huge uptake of nanography, so you can evaluate where they are located exactly. But you see in 2D dimension, you don't know if they are around the nucleus, or they are inside the nucleus, for this kind of nanoparticle, for vectors, for drug, and so on, or the possibility to see, for example, lipid droplets. This is another possibility. Usually by SEM or TM are visualized, but we are able instead to visualize and to localize all the lipid droplets inside the cell. So that means that, again, by labor-free technique, we are able to visualize all the substructures inside the supercompartment structures inside the cell. You can see in different conditions and so on. Again, about liquid biopsy, in this paper we try to use 3D information just to make phenotyping, just to detect a tumor cell against other cell that we mix them together just as a trial for these kind of things. And other phenotyping again by 3D instead by 2D. Of course, you are adding more information in 3D, and so it can help. And eventually you can just phenotype the object, and then you can have also the structure, how is physically the structure inside the cell. And so just to end in my presentation, I will give two examples. One, acute myeloid leukemia, very same paper, and another lysosomal storage disease. I will show you here the first one. I go very straight. I expect that you know about this pathology, of course. And we demonstrate the possibility to detect this strange shape, 3D shape of the nucleus in this case, this cap here. And so essentially we were able to make a correlation between genotype and phenotype. This is induced by the genetic modification in this case. So we changed our result about photonics, our strategy to detect this kind of nucleus here. You can see just the result in which the accumulation has some specificity just to detect essentially the correct shape of this nucleus that otherwise requires standard biotechnological things, by characterization, some features, and so on. So essentially we were able to detect quite accurately the shape. So we just by a quick analysis inside the microfluidic channel. The other side that we in collaboration with the biologists to the Teleton Institute, they gave us some Nima-Pick cell. Okay, Nima-Pick cell, as you know, wild type and this knockout. Okay, in which there is accumulation of cells. This is right in anticipation of the result that I will show you more detail later, just to explain what's happening in two different cells with the lysosome lysosome accumulation in this case. And let me go to show here how we are able to detect exactly which is the real distribution of lysosome aggregates around the nucleus in different conditions. And you see, we are able. So, if we compare how we can see this one and how the biologist in this Teleton lab is able to see by confocal microscopy, essentially it happened things like this. Due to the adhesion process of the cell, you see something that is modified, or there are some artifacts. Instead, we use in three-dimensional shape. That means that we are less subjected from the position that the cell can assume on the substrate. And so we were able to detect, to extract some biomarkers that are more general because we are using the cell every time in the same condition, in suspension mode. So we really know this one. But so we were able to discover these biomarkers. I will jump this. But the interesting thing was that we were able also to use some drugs and to make the circle around just by knockout, putting the drug, by making modification to see what's happening in the different compartments of the cell. I will show you here the movie. You can see real experimental data, and you can see from wild type, and so with the different conditions it's possible to going back and forth by cyclodystrin and other things just to restore the condition. For example, we discovered that the accumulation lysosome was restored, but the deformation was not. So that's the problem that can have some question, arise some question about this. So essentially now we have accumulated about 30,000 tomograms of cell, many cell lines. And so now we have data just for training and just for simplify all the process by AI, because we can feed them a neural network. What comes the next is just to use this for spheroids, or maybe for embryos, and improving the spatial resolution. We started also a spin-off, names last January. We were finalists in SPI meeting and as the last few slides, I will show that we are building also a metaverse. If we are building up a three-dimensional visualization like a tool for research, the researcher cannot see just on a flat screen this result, just to appreciate and also to discover new way, new strategy to for more accurate representation, visualization, and structure of the numerical value. Now you should work in a three-dimensional environment, and so with the help of some people, we published a couple of papers. I will show here one of the example. This is a virtual 3D. You put the goggle, you are inside the cell kingdom, and you can interrogate all the cells that are flowing along the microfluidic channel. And when you interrogate the cell, I skip a little bit, otherwise it takes time. And so you can go by teletransporter in this 3D environment, you can go enter in the cell, see what's happened, and maybe in the embryo or in the spheroids, to see a loop in proximity what's happened, and quantify, interrogate the system for interrogating for quantitative parameters that can be appreciated for this. We refined that work on a small journal on this paper that has appeared just a couple of weeks ago, and here we show you can see how we applying a more sophisticated strategy. We were able to separate all the subcomponents in this case in different conditions. You can see monocyte in this case with the vacuolin, different vacuoles, or we believe that in this case we were able to capture also mitotic spindle of this cell in different conditions, like with the nucleus and so on, or ovarian cancer cell again, and again by three-dimensional analysis for visualization and appreciation of three-dimensional analysis. In this case, in which you can integrate, separate, and compare directly the object, the shape, and morphology to see dynamic evolution of the process, and so on. So. Okay, just acknowledgement for some of the project. The main team leader, Lisa Miccio, Pasquale Memolo, Pietro Bianco. This my group in Munich 2 years ago about this. So thank you very much for your attention.
Thanks. Questions, please.
Thank you. That was wonderful talk and also very instructive. I was curious about the sensitivity and the resolution, both on the spatial scale and on the refractive parameters. And also, can you discuss a little bit on the smoothness of the models you introduced into the UI and
A lot of question in the question. Okay, the first question is resolution. We are the same resolution of standard optical microscope. Because we are using standard microscope technique. We are using somehow we are using also coherent lines. So that means we are also of course, I can know that digital photography there are some strategies that past improve the solution. we were thinking about that, but essentially, let's say, only just the standard was the
refractive index.
the refractive index. Refractive index we are the full picture change. Of course, if all the super-compartments they differ very easily with respect to cytoplasmic, so you should be very sensitive. That is one of the main problems. The problem is sometimes some people use strategies to change like chemical sugar, for example, by changing the soup compartment just to increase, but again, it's not a good thing. So the challenge is try to extract from the tomograms and definitely as it is the thing. So of course, now we are working also for challenging of mitochondria and different things like it's a work in progress. It's not the smoothness you see.
Yeah.
Because you have seen also, okay. So this is just for representation here, just to explain afterwards, just to give of course, that depends on the spatial resolution. So the minimum box that you are able to represent that depends on the spatial resolution. Of course, in GP, a little bit more complex.
Just to give you an insight of what I thought. You were showing an image of very large cells, which differ from the regular type. So I assume it was something like zinc cell, red cell. But also I was curious to see the same type of representation of the concave seeing. So having always a gap and also very spherical shape on the other side.
Yes, the cell is a renal cell with diseases. And of course, there was no time to show that a lot of measurement for the regular cell, a heavy regular cell also and okay, for regular cell, the machine is it's tricky, a little bit tricky application. But it was interesting because we were able to retrieve rapid effects again into the aberration here. Z90 it is again a Nobel Prize. After he got the Nobel Prize is studies people model of aberration that are usually studies are used for fulfilling the rights of our system. They prove the width passing through the optical lenses, and then one of the most hardest way to model is the natural one because they are photo terms, and so we are not detecting which one will be in the series. And this is important because we consider the retinal cell like a sort of aberrated lens. And when this lens is just in a tilted position, tilting is an aberration terms in the micro. It's an aberration. Okay, that workshop testing are testing here. And so by retrieving the aberration of this we are able to retrieve the end. So it was very nice because we are using mathematics of aberration to retrieve the end of these straight things. Also,
Can you just go back to the question?
It's probably might be more caffeine here, but for me it looks like you are still only using the face information. So it's 3D QPI, right? So what is the reason for doing that and not photography using both the information of the amplitude and the face? Would you improve, or is it an inherent advantage in what you do?
That's a good question because this morning I said maybe I have to present the slide which you say over 10% of amplitude. Of course, because it's familiar with it. Thank you very much. Essentially, amplitude, of course, we can also use an amplitude, but in amplitude we have no sensitivity. When you have essentially the amplitude in digital holography is the analog of the bright field image. You have some possibility to processing. You can have amplitude, you can square and have density, you can do something, but essentially it's bright. And so if you are not, it's not possible to see right if you are looking at face, because space is not the sensitive part of the whole. But you say, why you are using holography? You are using holography because of 3D picture. You know, our here it means that the fluorescent, the magnetic fluorescent you have to use hydrodynamic focusing of the cell. It's a complicated geometry for fluid dynamic engineering, just to be sure that all the cells flowing exactly in the line. Because if the cells are not flowing all to the along the single line, are just moving a little bit along the optical axis, you have a signal that is different, and you can have another. And so this signal, usually in the fluorescent microscopy, you are quantifying this signal. So if the signal is not correct, if the cell is not fully exactly the same position, they're not correct. In our case, thanks to the feature of holography, even if once the cell is flowing not the straight direction, this way parallel orthogonal to the optical axis, but the result it doesn't matter for us, even if they are doing here, because we are able to calculate numerical refraction integral and just putting focus everywhere. This is the magic of the holography. So we are emulating in computer the diffraction integral reconstruction. And so we are, for each one of the cells, we detect exactly which we emulate this one. It has just to make a focusing, which is its perfect focused position. And then we adjust all the position of the same cell at the same distance thanks to the holography.
I'm missing something. Because in holography principle, you have more information than you have in just in QPIs. You could do the same thing by recording the hologram. So why are we?
No, we got the deal. And we reconstruct the deal. We got the deal. I'm trying to explain how we use this order. Because essentially, when you reconstruct the measure holography, you can literally calculate what is recorded. Right? Right? And that doesn't matter because you can start putting focus this one this. Okay, putting focus this one is closer. Okay, you can. We are using this feature just to align everything and to have the same position of the cell with a different angle. Just
how long do you have to record the cell phone to create? Because you have really nice 3D constructions.
Yes, essentially the main aspect of the technology should have at least let's say 100 rotated holes inside the field, because of course if your field is limited, you cannot capture the field orientation. So this is the compromise that you have to. Because we should have at the end, depending on the angle of rotation itself, but we are able to engineering is so this is
it's a very impressive method what you presented. My curiosity is if you ever tried it on normal particles, not on the cells. So that the silicon particles with some inclusions inside, something that you can then break apart and see if it's looking like your image.
yes, thank you very much. Very interesting question because not on silicon particles, but because we have on polystyrene particles.
Yes, as good as.
And but we have done also on algae.
Polystyrene with inclusions.
But also all the
I'm
animals.
asking about polystyrene with inclusions. So you have small nanoparticles distributed in the side. Then you look at your image. I destroy the particle and I look at my image, and then I say, well, they look the same, or they don't.
Yeah, absolutely. It's true. We can if the object is transparent.
But did you try this? Because I know some particles which would be
we
interesting
try
or
a
not.
different object, like it's a silicate. The object item, the item and object is this animal that we put in ocean, in water, everything. The past they use it for our toothpaste.
Right,
And the external ingredient is in silicon. And inside there are two elements. And so we demonstrated in this paper, together with the rest of the site in 2017, we demonstrated the possibility to make homogram is just by visualizing the external layer and the internal layer. And this is very important because right now we are working on the possibility to measure chloroplasts
Right.
because they depend from pollution by human.
My question is much more straightforward and simpler. For a very simple shape, which is a spherical particle, with some very simple shape, which are inclusions inside, something that again, afterwards I can break apart and see. Is it possible to apply and is it working?
Is it possible if the object is transparent that you can see everything inside?
Silica is transparent, right? Okay.
Absolutely, yeah. That depends upon the size of your particle. If your particle is 200 nanometers, that's
No, one micron, 2 microns with nanometers.
absolutely. If you have to propose some experiment together, this has okay, to do very well.
Same thing.
Thank you.
Any other questions?
I have a question on your 2D part where you mentioned that your approach is able to recognize drug-resistant cells. So my question is, how did you test this? And then do you know which of the parameters the AI recognizes in the cells detects that this is the
Different class of the cell. Essentially, we put in machine learning. It is usually there are lots of information now AI for using microscope using bacteria, fluorescent analysis, and many parameters are usually density, some geometry, circularity, size, density, shaping different ways and so on. We added to this whole range of parameters that usually use also these quantitative things parameters. That's very important because it will be an addition, an important addition to all the set of features that are usually used in machine learning for verification. So we believe that one of the key success that we have added this kind of information. That's it. Now, for example, we have ability also to, for this one, also fractal recognize microclasses for distinguish between classes or diagrams again, just to phenotype their effect of evolution. So, of course, if you increase the richness of the features in your machine learning analysis, you have more chances to get. Of course, you have to add features that will have some part of the content.
So it was a supervised, a classified approach, and not the unsupervised, and you didn't know what did.
I guess just one short thing, because you're hitting the biodynamics, okay?
Yeah.
So, could you actually record something that like the spindles of the cell division, that you can see how they grow, or this is impossible? Because of the recording speed and the speed of the growth.
Yeah, this is just
Or basic
this
observation.
is a shot for a cell that occasionally pass through in that condition. So we saw some spatial temporal phenomenon. But essentially now we are trying to do something. Because some of the cells, the expression, you see some evolution dynamics and in a full because of course there are also potentially available systems to find models. There's a lot, but there are mainly for studying analysis, not really for cellular. And essentially, we are using the possibility to make a multiple passage for the same cell and so you fetch the just by making multiple movements of the very same cell passing multiple size there.
So could you ever see the spindles of during the cell division or so to catch them? Did you ever see that?
I don't know. Yes, I don't know.
You have to
The methodology is that if the division of the cell can be suspended cell.
some of the tumor cells.
Of course, if you have ionic fire, you need some also the problem is just single cell from one side, and on the other side is fast. For example, we are working on a sheet for and they are aggregates. But when you have aggregates, it's more challenging the rotation of the cell inside the flow. But we are doing something. And we are starting also about the steroids. We already started, and then there is a good way in Hamamatsu. In Japan, they have the similar things starting. 100 microns steroids. With the types of the cell size, they detect all the physical personality, measure activity of such kind of activity. So of course the degree of complexity toward the aggregates is challenging.
Yeah, very interesting. One of your objectives is high throughput, right? Sorry? One of your objectives is high throughput. Yeah. You have to take 100 images and you have to physically rotate the cell, which is not a fast process, plus very computationally intensive models. Is that realistic? And how long do you need to actually get the right
question? We are using parallelization to read and write a similar flow line of the cell. We can also certain very short side figures, so we can affect. So also the bottleneck is just speed up the process of the positions. Of course, at the end, if we compare the standard
Very interesting discussion. I have also plenty of questions because I was monitoring the reactions of players to the drugs and so forth. But unfortunately, time is very
sorry.
We can skip the lunch.
Thank you so much.
Any other questions? Good. So let's assemble this together again.